Amino acids, proteins. Proteins are compounds consisting of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Proteins carry out work in an organism. They are the machines, the enzymes, the transporters. They also form some important structures. Proteins are also called peptides and polypeptides. Proteins are chains of amino acids. There are 20 amino acids used to construct the vast majority of proteins. While there are a few others that are sometimes used, these 20 are the standard amino acids. All life on Earth uses virtually the same set of amino acids to construct its proteins. Amino acids always include an amino group, a carboxyl group, and a side chain that is unique to each amino acid. The side chain, sometimes called the R group, determines the unique properties of each amino acid. Here it is symbolized by the letter R. The chemical bond that is formed between amino acids is called a peptide bond. Like bonds between saccharides and nucleotides, they are formed by dehydration synthesis. This shows formation of a dipeptide by a dehydration synthesis reaction. A peptide chain with about 50 or more amino acids can form an individual protein. The largest known human protein, Titin, has 34,350 amino acids. Shown here are the 20 amino acids from which all proteins are comprised. Also shown are the abbreviations of each. Shape is critical to the function of a protein. A protein's shape depends on 4 levels of structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is the sequence of amino acids that comprise it. Each protein consists of a unique sequence. So, for example, alanine, leucine, serine, lysine, or valine, leucine, leucine, alanine, or lysine, alanine, serine, lysine, and on and on. Changes in the primary structure of a protein are changes in its amino acid sequence. Changing an amino acid in a protein changes its primary structure and can affect its overall structure and ability to function. Sickle cell disease is an example of a single amino acid defect which changes the shape of the hemoglobin protein. Sickle cell disease is a blood disorder specifically involving hemoglobin, which carries oxygen in the blood. A single glutamate amino acid is replaced by a valine in the primary sequence. The result changes the overall shape of the hemoglobin molecule and does not allow it to properly carry oxygen. Secondary structure is a result of hydrogen bond formation between amine and carboxyl groups of amino acids in each polypeptide chain. Depending on where the groups are relative to one another, the secondary structure takes the shape of an alpha helix or a pleated sheet. Note, R groups do not play a role in secondary structure. This is alpha helix, and these are pleated sheets. Tertiary structure is the overall 3D shape of the polypeptide. It results from interactions of R groups in alpha helices or pleated sheets. R groups, cluster due to hydrophobic and hydrophilic interactions, attract and repel one another due to charge interactions.
All right, let's talk about the quaternary structure of proteins. Some proteins have what's called a quaternary structure, which consists of more than one polypeptide chain binding together due to multiple bonds and interactions between chains. Tertiary and quaternary structure are often stabilized by covalent bonds called disulfide bridges, which you can see in the image here. We have this bridge between neighboring proteins that have 2 sulfides. They're called disulfide bridges. They form between sulfur-containing amino acids in the same, here we see in the same peptide or in different peptides. The structure or shape that a protein takes determines its function. So the function of a protein is determined by its shape. That would be its tertiary or quaternary structure. Protein shape is driven by amino acid chemistry. Protein function is determined by its shape and its surface chemistry. Each sequence of amino acids folds in a different way as each amino acid in the chain interacts with water and the other amino acids in that protein uniquely. For instance, upon contacting water, a protein could form a groove with an arrangement of electrons that functions as a binding site for other molecules, which is what you're seeing here. The way that this folds gives us this special binding site. And that folding, that shape, is determined by the uniquely arranged amino acids in the protein. So the key here is the structure determines its function.
Proteins have thousands of different roles in an organism. 8 common categories of function are shown below. Structural, hair, cell cytoskeleton. Contractile, as part of muscle and other motile cells. Storage, sources of amino acids or ions. Defense, antibodies, blood cell receptors. Transport, hemoglobin, membrane channels. Signaling, hormones, membrane receptors. Enzymatic, regulate speeds of reactions. Machines, motor protein moves organelles, microtubules move chromosomes.